But okay. Oh. The problem is that my daughter is having homeschooling here, so I can't participate in the session without the headphones. So
Yeah, I understand.
Yeah.
no. Sorry for that. No, your head will be the only fancy hat in the session.
Okay, well, that's good.
Are those leishmanias behind you?
Yeah, this is my backdrop, Leishmania amastigotes.
That's pretty cool.
Very nice portrait. Yeah.
Are these fluorescent or something like that, or?
They're for selfies. For people take selfies. I've got another one with a microscope. It's me looking down a microscope. So, yeah. So I've got a choice. It could either have the amastigotes or the microscope. So I chose the.
So this is from the exhibition.
This is from my parasite seeing exhibition. So it's a mobile selfie board. So
that's pretty cool.
Oh, amazing.
Is there some background noise?
Yeah.
Josias has the background.
People have a microphone or not. So we're expecting many people, or we've no idea?
Around 50.
Okay.
Yes.
Oh, some people names I recognize.
Yeah.
I shared the link with Richard Samuels. I don't know if he will have the chance to participate with us, but
yeah, I sent him a WhatsApp about an hour ago.
Okay. Yeah, I sent it 2 hours ago.
So I have a Colombian colleague, James Montoya Lerma. He's joined. He's a sandfly expert.
Thank you for the invitation, Rod. Nice to see you.
Yeah, nice to see you, James.
Nice to see you. Nice to have you join us.
So I'm just topping up. Are you waiting for me to start, or we just sort of?
No, I will make a brief presentation.
Right,
and
so
then
we'll wait until a few more people arrive.
Wait.
Hi, Yara.
Hi, Hadi.
Janta.
Hi.
I think you have to speak a little bit loud.
Okay. Can you hear me now? It's better? It's
Yes,
better now?
yeah.
Yes, it's not.
Okay.
Play.
Hi.
We're almost 40.
Yeah. Luciano, boa tarde.
Hey, Luciano,
Good
tudo bem?
afternoon.
Hi, Andrea.
Andrea.
Boa tarde.
Tarde.
Ô, Marcinha, tudo bem?
Márcia.
Adri também. Ô, Adri. Rod, we are saying hello to people that we don't see a long time ago. Oi, André.
Luciano.
Bom, boa tarde a todos, né? Ficar dando boa tarde para todo mundo vai ser...
É.
Boa tarde, gente.
Saudade de todo mundo. Não teve Ilha Grande esse ano?
Não.
Pois é.
Boa tarde aí para todos.
Mas tem um cheirinho. Assim, dá para sentir. Yeah.
A expressão é meio curiosa, viu?
Ou aquele que mora em Niterói, né?
É.
Ah, então tá explicado.
É.
Temos os 50 já.
Sim.
E não esquecer que o palestrante é britânico.
Boa, Luciano. Gente, é com você.
Luciano, o fuso horário é carioca hoje.
Você comanda. Não, o Genta é o comandante hoje.
Quer começar? Como é que você quer esperar mais um pouquinho? Qual...
Temos 50 já pode começar para não.
Tá bom.
Acho que podia.
Quer começar já então?
Sim, tá faltando
Bom.
o Pedro ali, mas ele deve estar vindo.
O Pedro confirmou ou teve algum rolo?
Não sei, deve estar no trânsito.
Ele tinha alguma coisa, ele falou que ia chegar em cima da hora, né?
Uhum.
Então vamos dar 2 minutos então, vai. Não vai matar ninguém.
Não,
Rod, we are waiting 2 minutes, okay?
Beto,
Well,
now is Richard entering.
Eu acabei de ingressar no doutorado.
Barbara. So many people.
You can see you still popular here, Rod.
Yeah.
Mr. Samuel is just connecting.
Yeah.
Lucas is here too.
Yeah.
A UFRNJ. A Coral Arthropédica, isso. Para mim, agronomia lá.
Moyashi,
Sim, o mestrado eu fiz com...
Bom. Bom, vamos começar então?
Fica à vontade.
Tá gravando já?
Sim.
Bom, boa tarde a todos. Eu vou apertar a tecla SAP, tá? Porque o palestrante é britânico. Well, welcome everybody. Rod, I will start, okay? We are already recording, so I will make a brief presentation and then you can start. Well, it's a great pleasure to present Professor Rod Dillon. Rod got a degree in biology in Brunel University, then a PhD in entomology in the University of Bath, beautiful place, and had was research fellow in the National History Museum in London, then research fellow in the University of Bath, and stay for some years in the Liverpool School of Tropical Medicine, and now he is senior lecturer of Lancaster University.
You missed out London School of Tropical Medicine, though.
Oh, sorry. I'm reading the CV you sent me. Sorry.
Sorry.
Thank you. Thank you for that. So, well, London School of Tropical Medicine, that was after Bath or before Bath?
Between.
Between. Okay. Sorry for that. Well, anyway, it's Rod Dillon. It's important reference in the entomology field. It's difficult to have a proper record publication for Rod because it's so many acronyms. But anyway, I got a record of more than 50 very nice papers, important contributions to our field. Some of them are very seminal, and to make a I think, a good description of Rod's role in our field. I would say that Rod was a guy that studied insect gut microbiology before it was cool, you know. So he laid the path for many of us studying the microbes of insect gut, and especially in vector insects in the last decades. From a personal point of view, I would say that Rod is one of the best persons I have ever met in science. We met in the 90s, a long time ago, and it's, I think, a good symptom of a good friendship is that the time stands still when we talk to each other, and this is very fortunate. It was a great luck to know Rod. We met in the University of São Paulo, and it was always a source of inspiration to be able to talk with Rod and to have contact with his brilliant ideas and brilliant discussions. And I got two important lessons from Rod in these years: that it's possible to do great science and being a nice guy at the same time. This was a very important lesson I got from him. And another one is that it's most important than the place you arrive is the friends you get in the travel. So I'm very glad to make this presentation. I'm very curious to see what you have to show to us, especially and one thing that is very important to say, Rod normally is a guy that is how can I say, anticipate several tendencies, you know. So it's very good to see what he's doing now, to see what will be important in the next years. And I think now his connections with the field of public engagement and art and connections with several fields teaches us that we should invest in those connections. And I think that doing multidisciplinary research and transdisciplinary research is something that Rod is doing for several years ahead of us. So it's a pleasure to present you, Rod. And Rod, it's up to you now. Thank you very much for joining us today.
Okay, thank you. So, wow, thank you very much, Fernando, for such a glowing introduction. And I hope I can live up to those expectations. Fernando's given a very good sort of preliminary introduction to what I'm going to be talking about. So first of all, I must apologize for not talking Portuguese. It's all Fernando's fault. When I first met him in Sao Paulo, I said, I must learn Portuguese. And he said, oh, we haven't got time for that. Let's just talk in English. So that's how it went from then on. So, okay, so let me share my screen.
It's shared. Okay.
Okay.
Yeah, it's perfect.
Fine. So you can hear me, everything's fine. Good.
Yeah.
So, yeah, I sort of plucked a title out of the air, really, sort of journey through the world of insect gut microbiota. So I'm into journeys now, as you might see throughout this talk. And on the screen here, you'll see my two sort of main insects that I've worked with through my career: the little and large of the insect world, the Schistosperma gregaria, there's a locust, and the Tamaya sandfly. So yeah, I'm based now at Lancaster University. The university, it's a top 10 university in the UK, in the northwest of England, and down here we have some of my sponsors that have sort of provided us
Congelou aí.
talking about historical perspectives on the importance of gut microbiota. And I'm going to sort of give a few sort of pointers about my journey through the subject and some of the choices that can be made and the crossroads that you can sort of have in your work and career. I'm going to be focusing mainly on sandflies and transmission of leishmania, and obviously leishmania, as I think most of you know, is a causative agent of neglected tropical disease. The second part of my talk is going to be talking a bit more, thinking about creativity and engagement, and the idea of we shouldn't always be comfortable in what we do. We need to be uncomfortable, and something called, what I call, is the discomfort zone. And something about talking about perspectives in our work, how we can encourage creativity in our researchers. Do we encourage, do we teach creativity? Do we nurture it? I don't think we do enough. The importance of play, the importance of public engagement now, and how do we engage with the public? I think emotional engagement is an interesting route into public engagement. I'll be talking something about that. Okay, so I love to sort of think about this quote that I sort of came across when I first started out really as a PhD student by an North American insect microbiologist working in the 1960s, Ed Steinhaus. And he said that a comprehensive understanding of the biology of insects requires that they be studied in an ecological context with microbes as an important component of the system. So that was a very sort of far-sighted comment for the 60s, I think, when you think about the total context of entomology and entomological research. And so from 1980, so I'm giving sort of, yeah, I've been around a few decades really. So I first started working on locusts, Schistocerca. I developed a method for rearing germ-free locusts, and there's probably virtually no other germ-free insect work research being going on at that time. And I started straight away to look at the protective effect of gut microbes against insect pathogens in locusts. And at the time, I shared a lab with plant pathologists, and this is one of the things I want to sort of allude to as I go along, is some pointers to promoting ideas and cross-activity, cross-research disciplines, is working and sharing a lab with plant pathologists as an insect pathologist, someone working on biocontrol agents and microbes. It was quite a productive thing, and I learned straight away that you could have some very good and useful conversations with people that didn't apparently have a great deal in common with you in terms of the subject matter. So one of the things I'm going to sort of be, the thread through this talk I'm going to be having, is that we shouldn't just stay within our comfortable disciplines. We need to go out and share and interact with other disciplines. This is very important, I think. So this is the sort of germ-free notobiotic system that I used. And I'm not going to go into much detail, but I showed that gut bacteria within swarming locusts create elements of the swarming cohesion pheromone, and that was quite an important step forward. And I think it showed, again, also showed that gut microbiota protect and create a healthy insect that can be, and that's important in terms of swarm behavior versus solitary and gregarious insects. So I think that was quite painstaking research. It took a lot of effort to produce these germ-free insects, but on the other hand, it was a nice large insect, a few centimeters long, easy to dissect, easy to work with. But funnily enough, at the time, in the 80s, I thought, wow, it would be good to develop a germ-free colony of Drosophila. And my supervisor at the time, I said, why don't we start working on germ-free Drosophila? And he said, no, we need to get on with the work that you've got the grant for, just get on with it. And I didn't sort of at the time push that idea. And sort of in some ways that is a regret, because of course 10, 15 years later there starts to be a lot of important work on germ-free Drosophila and the immune system and everything, and I could have sort of started up that field very much earlier on, but I didn't. So these are choices you've got, and something you need to bear in mind with regard to what you do in your work. So then I moved on to work on sandflies. So it's quite interesting working on a neglected tropical disease in the UK, particularly sandflies. Was it the best career move? I'm not sure. I think it was, but yeah, that's where we are. So I think, I'm not sure how aware the whole audience are about leishmaniasis and sandflies, but seeing as this is going to be sort of disseminated later on, I'll give a couple of pointers about and reminders about leishmania and phlebotomy in sandflies. So leishmania infections occur, sandflies are vectors for leishmania protists, and the main thing of interest for me at the time was that leishmania caused gut infections in these insects, and this is a DS red stained cohort population of parasites within the gut of this adult female sandfly, that's created by Hector Diaz. And so it's important because I was interested potentially in what happens with the other microorganisms in the gut of sandflies versus leishmania. So what is leishmaniasis? I don't think I'm going to sort of bore you with that sort of background information, but for benefit of the wider audience, I'm putting this slide in here. It's quite an important neglected tropical disease, and the main point is that it's transmitted by these tiny blood-sucking phlebotomine sandflies. Natural reservoirs, loads of different reservoirs, including humans and dogs, and humans are infected. So in terms of, I became interested in ways to try and prevent transmission of leishmaniasis. And of course, we know that in order to tackle these vector-borne diseases, we very much require an interdisciplinary approach. And this approach probably is provided here is the sort of traditional set of sort of disciplines that will be working together in order to achieve some sort of disease reduction. So drug development, vaccine development, but I was particularly interested in disruption of transmission via the sandfly vector. And more latterly, in the last 10 years or so, I've particularly become interested in how the community will participate with us and with other stakeholders, if you want to say that word, in reducing the disease and disease transmission. And community participation and engagement is the key point, because we can have the best best ideas in order to reduce transmission in the world, but if we don't bring the public along with us, we don't get community to participate with us, then the best things in the world just won't be able to work. So when I started working with sandflies, actually with Richard Lane in London School of Tropical Medicine in the 90s, my immediate thought, well, there must be an important role for gut microbiota in sandflies, because when you think about sandflies and how they live, they've got a unique set of features that means that the microbiota must potentially influence leishmania transmission, because of course leishmania stay within the gut during their residence in the vector. Sandflies feed on plants. That interested me because I used to work on plant feeding insects. It's only the female that feeds on blood for egg production. And of course the larvae living in soil, feeding on a microbe-rich compost material, and these potentially could be transmitted via the pupae to the adult form. And also the female sandfly is a pool feeder, cuts basically a wound in the skin as a bit of a messy blood feeder. It doesn't have this neat sort of feeding process, so there's a potential for contamination of the blood meal as it's taken up by this tiny female sandfly. When I say tiny, some of you might not be aware, they're only sort of a couple of millimeters in size. So they're not as easy to dissect as a sort of large 3 or 4 centimeter locust. They're quite dear to my heart. So there's a bit of historical context really about sandfly research and recognition of microbiota's central role. I guess sandfly research has been going since the 1920s through to now, but up till the 1990s, I would say that there was no interest whatsoever in terms of, apart from one paper, some work by Schlein and the Israeli group, I guess, in the 80s, there was no real work, sort of idea that bacteria could be sort of interacting in some way with the leishmania. In fact, one of the leading sandfly experts in the field at the time insisted that the sandfly gut is sterile because of the blood and that there was no other sort of bacterial interference. That's what my sort of response I got when I started to engage this expert about my sort of ideas. So it's not really, so we did a study on gut microbiota in sandflies in Egypt in the later 90s. It wasn't really until the 2000s that gut bacteria, the idea that gut microbes in insects could be important started to take hold. And then I started to see a few heads popping up over the fence and having a look, see what was going on, rather than sort of ignoring me and sort of passing the other side of the street, as it were. So, and then I think we've seen the last 10 years, particularly the last 5 years, we've seen quite a sort of increase in interest and work being done on the importance of gut microbes, particularly in sandflies, but also in other insects that we're interested in, kissing bugs, mosquitoes, etc. So Eriki and Yara and others spent a lot of time and labor doing this network analysis of sandfly microbiota. And there's a lot of stuff that you can sort of take out of this. I'm not going to talk about it in any detail because time's moving on, but I think it shows you where we are now with regard to starting to get information about the characterization of the sandfly gut microbiota. Excuse me. And I think it's interesting, but there's still a lot of work to be done, because I think, how do you sort of differentiate between resident species and those bacteria and microbes that are passing through? What are the main players? And this network analysis comes some way towards sort of giving us some clues, but I think there's still a lot of sort of work to be done to really work out what are the real players going on. And there's been some work done in NIH to sort of give us a bit more information about that. But that's something I think needs to be still worked on more strongly. So we've got this model system that we've been using in my labs in the UK.
Hi. May I ask a question about the previous slide, Rod? Yeah. So, yeah, obviously I'm a kind of lost with so much names. So the simple question is, there are some dominant genera among those, so some other it changes also.
I think I'm going to sort of mention some of those dominant genera in a couple of slides' time, related to some work done in NIH. But I'd say generally that there's still a lot of confusion about what are really the genera. So we've done a lot of sort of collecting species that are present. What are the most important ones? There's some work being done in the last few years, but there's still a lot of work to be done, okay? Yeah, okay, so the model system that we're using with Infantum Shingazi also permits the use of infection with Leishmania mexicana. And we use this system, membrane feeding system, that a lot of you are using, I guess, with mosquitoes or bugs, etc. So I'm not going to go through in any detail, but you know, one of the questions that I have been asking for a long time, and I eventually got the funding to work on this, was, does gut microbiota interfere with Leishmania development? And this is what I call sort of colonization resistance experiments, and I'll sort of provide a couple of slides here, but basically the take-home message from these slides is that in the presence of certain bacteria, these bacteria are actually isolated from the Tsamaya in the field in Terrazina and brought back to the UK. And so basically the message here is that in the presence of certain bacteria, numbers of Leishmania population can be reduced, and there can be an additive effect of the different bacteria. And that leads also to potentially significant reduction in infection in certain combinations of bacterial species, but by no means all of those species. And so the other thing that I was interested in that project was the costs and benefits of leishmania to sandfly. We always heard a lot about the costs of vectors in carrying these so-called parasites, but I was quite interested in the opposite question. Are there potential benefits to the sandfly host in possessing the leishmania populations? Indeed, does actually leishmania protect the sandfly in some circumstances? This was an interesting point in my career in the terms of I actually applied for some funding to work on this question in the 2000. That's like sort of 12 years before I got some funding. And one of the reviewers actually said in response to my application was that my ideas were odd. And so I thought, wow, is that really, that really was sort of surprising to me, because I thought, because an idea is odd, does it mean it's not valid? So I think that sort of is quite a sort of important comment and reflected badly actually on the reviewer. And so that actually held back my research. I then went off and did other work, and then I came back to work on sandflies later on, and I was able to get the funding and actually answer those questions. So basically, I did some experiments, and I showed that if you infect sandflies with leishmania, you could then challenge those sandflies infected with leishmania with an insect pathogen, and then you could record their survival. And again, I'm just going to quickly sort of give you an overview of the result. That sandfly survival, where the sandflies had leishmania in their gut, they're able to survive significantly better than sandflies that were fed with the bacterial pathogen, in this case Serratia marcescens. So they were able to, as I say, they were protected to a certain degree, same as the controls. So basically, that was a sort of step towards addressing the hypothesis I had that leishmania could potentially, under certain circumstances, have a beneficial effect on the insect vector. And so you can see, you know, that basically there was a sort of trade-off and sort of benefit to both partners here, in that the leishmania had an increased chance of transmission, and the sandfly was more able to survive. And this could be potentially quite important to use, for example, of biocontrol agents, because it shows the complexity of the interactions between the players, the leishmania, the other microorganisms, and the sandfly host, because it may be that if you use a bacterial pathogen, you could actually knock out the sandflies that don't possess the leishmania and just end up with leishmania infected sandflies surviving. That's just a hypothetical sort of scenario. So there are other people started to get interested in working in this area, and they're actually addressing the sort of other question about are members of the gut microbiota in sandflies beneficial to leishmania development? And so Kelly et al. 2017 published the paper again from the NIH group. And here they were sort of showing up that, to answer Pedro's question here, the bacteriaceae were becoming dominant within leishmania-infected Lutzomitis sandflies, and numbers of Pseudomonas were diminishing as the parasite population increased. So you can start to get some detail of what's potentially going on with the interactions of leishmania and different bacterial genera. And the other important point that they showed in this study was that antibiotics included in the sandfly food were then caused the parasite growth to be depleted and metacyclogenesis of leishmania to be depleted. So, and it seemed to be a true effect of a reduction in the certain bacterial population, which seemed to support bacterial leishmania growth, then suggested that there were indeed some benefits in having certain bacteria in promoting leishmania growth in the gut. So we start to see that there's quite a complicated set of interactions going on here, potentially some beneficial effects of certain genera, some detrimental effects of certain genera, which is what you might expect, but we might have started out with quite a simplistic idea of what was going on. And I think the sort of story became more complicated when we started to think about, groups started to think about, okay, so there are bacteria in the gut of sandflies at the same time as leishmania. So what happens when a sandfly bites on a host? There are going to be leishmania and bacterial antigens being released into the wound. And what happens there? What happens with regard to the mammalian immune response? So that's what the study by Day et al. With Chardon, Kamhawi, everyone in NIH were working on. So they did a fantastic study. I'm pretty jealous, actually, sort of, yeah, great stuff. But they show basically that the gut bacteria transferred during the bite actually influences the severity of the disease process. So it actually shows that the gut bacteria triggers the inflammasome, amplifies interleukin, and sustains recruitment of neutrophils to the bite site. And so giving antibiotics to sandflies before transmission abolishes neutrophil infiltration, and that actually reduces leishmania dissemination throughout the body of the host. So wow, that's really starting to show another level of the importance of gut microbiota in the leishmania transmission process. And I think we're poised at quite an interesting point now about where we can go with this and how we can potentially manipulate the system in order to come up with some interesting new approaches to not only disrupt transmission by the sandfly, but also determine reductions in dissemination within the host. So there's a schematic cartoon about what they did. So this part of the talk, I'm just going to summarize now, really, to say that sort of it's been a sort of long road of sort of bit of a lonely road at the beginning. You know, I used to go to entomology conferences and talk about gut microbes. No one was interested. Then I'd go to sort of gut microbiology conferences where people were talking about pigs and cows and no one's interested in insects. So finally, there is an audience to talk about gut microbiota of insects, and that's great in the last 10, 15 years or so, and I think it's only going to get a more populated area. So I think the message is that microbial ecology, the whole ecological processes of sandflies sort of interacting with the macro microecology provides us with a sort of quite a daunting set of networks and complex networks with sort of interplays of negative and positive effects of microorganisms that are quite difficult to sort of dissect out. But I think we have to sort of look at that whole picture, and that's what I want people to do, to have a more holistic approach to things. And I think also the idea that leishmania are not just passengers, they could have detrimental effects in terms of egg deposition, but also that they could be potentially beneficial to the sandfly host is an interesting point that we could sort of look into also in more detail. Okay.
Before you change gears, may I make a comment and ask a question? Yeah. That comment is just to tell you that I teach parasitology at my university. Somehow I end up teaching it in the last few years. And the first class is to discuss what is a parasite. And one of the first figures that I show to the people is that your figure of the Leishmanian serration. Because then to change, what's a parasite at the end? At one moment, it seems to be a mild parasite, but at the other moment, it seems to be a mutualist symbiont. So then the parasite depends on the context.
Yeah,
that's it.
Yeah, thanks very much. That's very true. So I think that said it all, Pedro. I think we're sort of very focused on our ideas from what we've learned during our degrees, and it's quite difficult sometimes to break out of those ideas and have novel and new thoughts. And this is something that I'm going to be sort of talking about in second part of my talk.
Yeah.
I'm going to sort of around creativity and engagement, some sort of thoughts on this.
Yes, that's related. And so the question, what's your feeling about the mechanism behind that mutualistic effect on that? So it changed, the parasite is changing directly the microbiota, so it's an interaction between the bacteria and the parasite, or that interaction is mediated by the parasite pushing some buttons in the host to change the microbiota indirectly?
I think my feeling is that the leishmania is pushing some buttons in the sandfly that is sort of changing the immune response, and it could be peptides, it could be reactive oxygen, it could be a whole range of things, and it's sort of who can run the fastest, who can sort of withstand what is going on in the gut. And I think the leishmania tends to win out under those circumstances, but yeah, it's still something to be worked on and researched, but that's my sort of hunch. Okay. So yeah, being uncomfortable in your work. Welcome to the discomfort zone. That's what we're talking about. And then a bit about different perspectives, about encouraging creativity in researchers, and the idea of emotional engagement. So this was a cartoon developed by the Royal Society a few years ago now about the culture of research and the future of the science system. And I think for me, the interesting point was what's in the sort of middle of this schematic, this whole network of sort of processes to do with dissemination and research. The redesigned system is collaborative, multidisciplinary, open and creative. And that for me is the sort of where we need to be going, where we need to be sort of focusing on. Because I think, yeah, creativity, it plays an essential central role in science research. And we need to be, as researchers, highly imaginative, to set up goals, to find unexplored solutions to the research problems we have. And I'm also interested in the way that science can influence other groups to express their own creativity, and I'll talk a bit about that later on. So some of you might have sort of heard about this in the past, but I think the idea of play and sort of relaxation to nurture new ideas is something that we should sort of think about and take seriously. So we should take play seriously and not sort of discard it as something that's childish. Play is not childish. It's an adult thing. It's something grown-ups should be doing. So basically, I had an idea when I was eating my lunch one day. This is my lunch, very healthy. Some parsley, some sort of grapes, homemade bread, and some tomatoes. And I thought, okay, so I wonder if sandflies would feed on tomatoes. And that question, so then I said to Fernando, who was working with me at the time, I said, you think I should feed sandflies tomatoes? And he said, why, yeah, why not? And then he started to think, oh, maybe we should try to feed tritomine bugs on tomatoes. And that was quite a sort of astonishing, game-changing sort of thoughts that Fernando had, really, considering the whole history of Hodgkin's, prolixus and Chagas disease, and how they were considered to be exclusive sort of blood feeders. So that led to a whole new sort of, that idea of a sort of playful, shall I put cherry tomato in the sandfly cage? That idea led to a sort of basically a new research project. So I think this is the first of a Fernando picture. I've got quite a few pictures of Fernando in the archives, so I thought I'd bring a few out now and then. So I think we were in Chirk Castle on the Welsh borders when we were discussing, oh, maybe we could feed sandflies and tritomine bugs with tomatoes. And yeah, we know that creativity demands a bit of rest and relaxation. And here we see a rare occasion where Fernando is sort of resting and dreaming about his, I think it's not his first love, obviously, because yeah, his wife, but and kids. But the tritomine bugs are quite up there as a sort of in his thoughts, I think. So I've got a plea to you to sort of not always be comfortable in what you do, to challenge yourself. And this is particularly a case for the younger people in the that might be in the audience, maybe. So are you uncomfortable? then you should begin, because I think my concern is that the traditional career pathway of the entomologist and the vector biologist doesn't always provide us with the most creative of researchers. And that's something that we need to think about. I think we as researchers should be looking outwards for ideas. We shouldn't always be talking within our groups, but we should be talking to other people. There are specific other people that I can sort of recommend, but in general, we should look outwards for our ideas. I would encourage people who are in a career in vector biology or research entomology, if possible, to sort of take a break and do something else for a change, to change the mindset, change perspectives, and to always work across and with other disciplines. So in fact, I'm sort of, I would suggest that the specialist, the ultra specialist, is now sort of not always the go-to person, but that we should be looking at people who can specialize and generalize. So you can be both, if possible, to drill down and have some specialization, but also have the ability to generalize and work across disciplines to be able to master multiple disciplines at a certain level. And then we all carry within us the potential to both specialize and generalize. And the importance there with the generalization of working with across the disciplines is to then have people that you can work with, that you can trust, who are good in the other disciplines, so that you know just enough to talk to them, to work with them in their discipline, that they have the depth in their discipline to bring their discipline to bear into your work, to really do something special together. So I did that at Lancaster about 6 or 7 years ago. I spent 8 months working completely out of my comfort zone. I managed a group of sort of computer scientists, designers, and a homeless community. And we had the task to develop a piece of technology in 8 months. And we actually did that. This piece of technology, which cost a few dollars, is to help homeless people have some access to information. I'm not going to go into the details of that, but so well, what's the relevance of that? Why is that of interest to what you're doing? On the surface, it doesn't seem to be relevant at all. But the first point is that I learned that working with people who are designers, people who do design thinking, was quite a sort of interesting and unknown thing for me. And so there are these groups of people out there that we can work with that potentially a lot of us don't even know about, that can be very sort of productive in working together. So the whole design thinking process, designers who actually are ideas designers actually help you to work with you as groups to systematically extract, teach, learn, and solve problems that we have in very creative and innovative ways. And so I would recommend perhaps that you look up your sort of design community in your university or your institutes and sort of make some contact with them and sort of see what sort of conversation you can have. And actually from that piece of work with the technology, we actually published some publications on how you can actually co-design prototypes with vulnerable communities. And that led me to think about how we could introduce those concepts into working with communities who have these neglected tropical diseases. So it's all about relationships and sort of working in parallel with people, and also it's about how you can use other cohorts, other people like design communities to actually sort of give you a fillip and sort of move you in a different direction. And related to that, I think Ross Downsill is on this talk on this Zoom meeting today. Ross is doing a PhD with me. Ross is what you might call in the UK a maker. He's like a sort of artisan, freelance geek, if you know the term. So sort of he makes stuff, and he's outside the university academic community. He's got a very different background in electronics and making stuff, and he's working on a PhD with me and the arts faculty on something called critical kits, and he's actually at the moment sort of designing a diagnostic system, but is also doing things like this prototype sort of Petri dish maze for Drosophila. There's a lot of play and sort of open source learning, and I think that's another group of people that are very interesting to work with. So go outside of the academic community into other communities of experts where there's a huge creative pool of talent. And I think the Prakash Lab at Stanford, for me, is one of the top sort of examples of this, if you like. And I think he's done this whole load of sort of technology products, sort of what we call frugal science. And the most recent one this last couple of weeks was the production of something called the Bitoscope. I think that's interesting because firstly, it's a fairly cheap way of changing perspective on looking at mosquito behavior, but also the way that he's sort of bringing out this information. It's sort of very sort of engaging Bitoscope, but also it's imaginative, it's creative, and I think it's sort of one of the labs that I think is really doing some really interesting and creative work. So it'd be good to look up that. But time's moving on, and I'm just gonna sort of skip along a little bit more. And I think I want to finish off about sort of discussion about how we can engage creatively with the public. And I think one of the things that I've learned over the past few years is that getting up, giving talks is not always the best way forward. In fact, it can be a negative thing. In fact, it can put people off. It can send people in other directions. And so there are other ways you can engage with communities and the public, and that is through engaging with their emotions through sort of artistic routes, if you like. So this is really interesting for me. So this was some piece of work produced by a 20-year-old undergraduate in my class, microbiology class in Lancaster. And so they're asked to produce some piece of art with using some microbiological techniques. And so this student came up with this concept of showing how antibiotic resistance is occurring and how we're running out of time with regard to antibiotic resistance. And so if you know about bacteria, basically there's bacteria growing across this plate, and these antibiotics are killing off the bacteria. These antibiotics here are having some effect, but there's antibiotic resistance here. So this student sat down and designed this image. He had to have understanding of the role of antibiotics, susceptibility and resistance. He had to have the technique to do this. He came up with this in a couple of hours, 20-year-old student. So, wow, I think that's an interesting mark of, you know, you don't see creativity like this in students but you have to look for it. You've got to nurture it and bring it out. And so I'm just going to sort of finish off by talking for a few minutes. Have I got a few minutes? Yeah. About a set of projects I've been doing for the last couple of years called Parasite Seeing with my arts colleague, Jen Southern, at Lancaster University. We had funding through Wellcome Trust with the Wellcome Centre for Anti-Infectives at Dundee University. And we had the idea basically of inventing firstly a travel blog for Leishmania. And I don't think I've put the sort of, yeah, I think you can find that online. But basically it's changing perspectives again. So it's looking at the world from a perspective of Leishmania. So the digital online sort of project was basically to actually look at how Leishmania travels through the labs, like in Lancaster in my lab, how Leishmania travels through an anti-infectives drugs lab, how it travels in the world, in the wild, and how it travels through time. And this was accompanied through a lot of sort of social media feeds at the time. And I had sort of ideas about, you know, this is, for example, a sandfly looking for somewhere to lay its eggs. And then sandfly traveling around the field. And of course, there's a sort of, this is a sort of Leishmania traveling through Leishmania macrophages. This was invented by the scientists in Dundee University. So I also learned how to tweet as a Leishmania parasite. And that was quite an interesting process because I had to think about things like, how do you address a Leishmania? What's the gender? And so I did actually develop some quite interesting perspectives about Leishmania based on sort of these projects that I was doing with Twitter, etc., that I can sort of talk about at length, but not at the moment. And then we developed the airport lounge. So making the unfamiliar familiar. So it's taken something that's familiar that people know, the airport lounge, and it's sort of transforming that into the idea of Leishmania checking in to an airport and going onto the sandfly plane. It sounds ridiculous, the word trite. I don't know if you understand that, but it sounds, we shouldn't really be sort of making these things into a human anthropogenic sort of type of idea, but it seems to work basically. So Parasite Seeing, the departure lounge. This is your check-in where you get your passport for your Leishmania and you become Leishmania in the journey. And there's the departures board where you have your destination, could be a dog in Belo Horizonte, or it could be a sort of human in India, and the flight is Lutsamaya or phlebotomers or Gentipes, etc. And there is sort of a way to sort of engage with the Leishmania, the Twitter feed there, and there's sort of some pictures of waiting for departure. This was pre-social distancing, so it's quite interesting. But the whole idea of sort of barriers and sort of passports is sort of something that's become now quite relevant to spread of infectious diseases, obviously. And we even produced a free airline paper that could be sort of looked at and read before boarding. And we have the sort of boarding process. And then I even had the sort of leishmania LPG story through a sort of magnet attached to the ceiling inside the sandfly gut. I think I'm going to sort of leave it at that, but basically this is a final sort of thing that I did a few years ago. This is an art performance I did in Liverpool with my wife, a day in bed, talking about bed nets and sort of tropical diseases and why so much money is spent on warfare. And I don't know if many of you have seen this, but I'm gonna sort of, I might give you a bit of a shock, but this is okay. So I just want to finish sort of reflecting about, you know, where are we going to be in 20 years' time? What's a research entomologist going to be? Well, I think that idea has already sort of taken a bit of a change in the last 6 to 8 months. I think we've sort of surged forward a few years in what we're going to be doing. But I think we can also see the beginnings of an idea that a research institute or a university is not actually a place necessarily, but it's a process, a situation where people can sort of exchange ideas and network. But there will be, you know, artificial intelligence, computers, automated network labs. There's not going to be lots of technicians sitting there sort of with their pipettes and sort of test tubes and everything. And I think that the important thing for me is to think about this now, about the people and the scientists that we want to sort of develop, because creative thinkers as students and researchers who are going to work and play together with these converging strands of science, art and technology, with the people outside of university and academic communities coming together and the public as well. I think, you know, we've got to change our game really in terms of how we're sort of educating and creating researchers for the future. So I'll just finish off this sort of slide here. Questions about, what do you think about the second part of my presentation? Do you think it's irrelevant to you? Do you think of yourself as creative? I had quite a few surprising sort of responses when I asked people that question. And do you sort of generally, when you think about your insects, do you think about them in terms of from outside inwards, or do you sort of think in terms of how the insect might be behaving, for example? So with that, there's some of my collaborators. I thank a whole list of artists, scientists, makers, etc. that I've been working with over the recent years, and I'm happy to take questions. I'll Stop sharing.
So thank you, Rod, for the marvelous presentation. Somebody has any question or?
Tá muito baixo o seu som, Fernando.
Okay, okay, again. Can you hear me now?
Yeah.
Okay, thank you. Sorry for that. So thank you for the marvelous presentation, Rod, as usual. So somebody wants to make any questions? Or, well, we have one question from Leonardo Abreu in the chat.
Okay.
He asks, what sort of responses from the general public did you get with the parasite seeing approach?
Okay, so the parasite seeing, the departure lounge was actually, so that was installed in the life science arts sort of arts place in Dundee, and that was the best show, the most popular show that they've ever had in the life science art space, actually. So it was extremely popular, I'm pleased to say. The online digital arts parasite seeing is more difficult to gauge the people's sort of response to that. But I think this is what's interesting in terms of our discussions, perhaps, is that it was great as a piece of art, but it wasn't necessarily the greatest thing for public engagement. So there are subtle differences, and sometimes what makes for great art doesn't necessarily make for an educational piece. And that's also related to the idea about how scientists and artists actually interact with each other. Do they exploit, or is there a mutualistic benefit, or is there parasitism and exploitation? That's something that we're quite aware of in what we do.
Well, here, how do you get, a question from Yasmin Kumordzi, how do you get more scientists involved in arts or creative engagement with the public?
So I think you should start them young. So you need to get the students involved in sort of creative engagement processes. I know in Osvaldo Cruz, you have undergraduate artists working with different groups. And I think that exposes young scientists early on to different ideas and to different sort of questions coming from different perspectives. And that can be very refreshing. And it means that those young scientists have already been sort of brought into an environment where things are different and things are sort of more challenging and creative, because art students coming in to work with science students, art students will ask weird, sort of unexpected questions potentially, and that will sort of create questions in your own mind. And so I think start them young, get them involved straight away. And I think that's also, if we're presenting engagement to the public, we need to have people of all ages, not old guys like me, sort of with a beard and glasses, the classic. So you need to have young scientists sort of to do a lot of the engagement as well.
Now, a question from James Montoya. How did you measure the impact of your creative approaches?
Typical question from a scientist, that one, isn't it? Yeah, that it's sort of, so it's more sort of questionnaire type of approaches rather than, it's qualitative information rather than sort of quantitative, statistically sort of tested information. But I think it's sort of getting feedback, general feedback and questionnaires, that sort of thing, really. And people watching people engage, you know, the public engagement, people in Dundee, for example, as I say, they were very sort of impressed and able to say that had lots of people coming in and sort of engaging enthusiastically, and they did do sort of questionnaires and things like that. It's a bit outside my sort of personal specialization. I know a bit about that, but not very much. So I leave that to other people, basically.
Well, I have two questions. They are more related. They will be a more scientific perspective than James. It's about the first part. I was curious about if the impact of leishmania on the bacteria, on gut bacteria, is indirect. Do you think leishmania can have some, or any kind of parasite? We don't know, but let's talk firstly about leishmania. Do you think these parasites can have some impact on insecticide resistance? For example, messing around with xenobiotics response or excretion?
I think, yeah, I think potentially they could. And we sort of discussed some of these effects of gut microbiota as well, haven't we, on insecticide resistance? I think someone's doing some work on that, but yeah, we didn't get funding for that. But I think, yeah, potentially there may be, when you think of that potentially large population of leishmania residing in the gut of a sandfly, they could potentially have some sort of detoxification activity in addition to what the sandfly host is doing. It's a good point, good question.
Well, the second one is more historical. It's the knowledge about the gut, the knowledge about the interaction between microbes and insects is quite old. We have historical examples from termites, ants, kissing bugs. What do you think were the main impediments to extend this knowledge or extend this kind of research for other models, as sandflies, for example? Do you think the most problems were theoretical or technical? For making this research odd.
Yeah, I think when you think about how we sort of have our careers, we sort of in quite a narrow set of labs, and we sort of learn from an expert vector biologist, and it is quite difficult to sort of it was quite difficult to sort of bring in ideas and information from other fields. So you're right, there was a lot of information out there about the role of, for example, gut microbiota, but it seemed to be there are fashions, there is the sort of, with the horse you've got these blinkers, you know, sort of you just looking ahead, and there is the time element as well. And also, I think the referees as well, referees of grants. I'm sad to say, sort of not open-minded enough to sort of accept these odd ideas. And so there's a whole range of things sort of working against us, sort of moving more quickly, being more creative in the research that we do. And that, I think that's what sort of happened. I think I'm hoping that will change, and I think maybe in some ways this year with this unmentionable thing has sort of caused some sort of nurturing and sort of development of creativity in the scientific community. And I'm hoping that that's going to change as things go forward.
Well, there is another question from Angela. Why the clockwork orange look?
I can change the look if you like.
Yeah.
Well,
That's fancy.
you have to explain that this is not a mosquito face, you know.
No, this is a playing mask. For those who don't know. But next Monday, I'm giving my first year microbiology lecture. So I always put this mask on for my first year lecture. Usually it's in a lecture theatre and I walk around prodding students with this on, but now it's virtual, which is not quite the same. But yeah, I bought this in Venice a couple of years ago. So it's now become quite sort of topical. So you actually fill this with herbs. So at the time, there was this sort of conflict between the idea that germs cause disease versus miasma, smells. And this was in vogue when people thought smells cause disease. So it's the miasma theory. So, yeah.
Well, we have a comment from Ross to encourage more scientists. Rod and I thought about maybe starting an informal art and science club, maybe, and impact of creativity can be hard to assess. Artists could learn how to analyze the tests of their qualitative questionnaires from scientists, qualitative and quantitative approaches, like the network analysis of the gut microbiota, but analyze audience responses or content of informal discussions. So this is a contribution from Ross.
Yeah.
And somebody wants to make oh, there is another comment from James. It would be a nice idea to promote these kind of ideas in our countries where the transmission takes place. How easy is to bring them?
Well, Fernando and groups in Osvaldo Cruz are already sort of world leaders in arts and science, actually. They've been doing it for many years. People like Annunziata and now Fernando are involved. And so they are some of the experts in South America, so you should talk to them. And we're working together on some new projects as well, aren't we? So.
Pedro wants to make a question.
Two. One thing is about the art question topic, and the other about the computer, the PC for homeless. And about the art, I was thinking that we're looking for so the focus, almost the work you showed was more on a nice view of the insects. So they look nice, they look friendly, beautiful, elegant.
Right.
But that's not the starting point of the way most people feel and think. And so most of the people have a kind of, they are afraid, they panic.
Yeah.
So they have all types of phobic feelings about insects. So it's weird.
Yeah.
It's a weird thing. It's an uncomfortable feeling for most of the people. So I wonder if in the approach to the trying to produce art and to link with art and art with an entomology, at least the starting point should not be the uncomfortable.
Okay, yeah. So
because
comfortable
not the way we look.
Yes,
we are weird.
Particularly you.
So, we are the people who look for a mosquito and think, wow, what a beautiful animal.
So the uncomfortable point is for us, I'm asking scientists to become uncomfortable in the work they do and the people that they sort of engage with. But this is slightly different. So what I'm not asking the public to become uncomfortable. I'm asking them, I'm trying to engage them with the diseases that we're working on. And yes, the airport, the airline, the plane is a comfortable thing that everyone knows about. They haven't been on it perhaps, they've seen it on television. And that's a comfortable thing for them to then go into learning something about sandflies and leishmaniasis and not sort of putting their hands up at the beginning to, oh, I don't want to know about these things. So it's a process of, oh, a little bit of engagement. Oh, this is interesting. And then gradually they learn something about sandflies and leishmania. And then they go, oh, is that what they do? Oh, they do that, do they? So then it sort of creates a few questions and a sort of germ of an idea of they might understand something and not be put off and horrified. So yeah, you're making a good point. I'm not asking the public to be uncomfortable. I'm asking you guys, people, to be uncomfortable, or particularly young scientists to be uncomfortable at certain points.
Okay. Then the other thing was about the computer. So here in Brazil, during that pandemic, that strange moment that we are passing through, a lot of students lost the access to school, and a lot of schools were not able, felt that they were not, simply were not able to provide content and classes through the internet to the students. And one of the alleged reasons for that is true in part, it's not completely true, but it's true in a large part mainly for in several areas of the country, but even for a part of the students, even at the university level, so they don't have hardware and things like that. So what's the cost of that jigsaw computer you showed there?
Okay, so that was just a few dollars. So that was actually something that provides a printout for homeless people that they could find out about their appointments and everything. So they get a printout, a waterproof piece of paper that prints out emails about court appearances, about going to the pharmacy, for example, for their sort of treatment, drug treatment or something. So I think, yeah, it's a very important point about sort of computing and the cheapness of computing. But now, I mean, we're seeing with mobile phone, smartphone uptake that there is much increased access to things like smartphones. For example, I work in Ghana, and there's 3 times the number of phones as there are people in the population. So I think technology is becoming cheaper, and the electric sort of power source through solar power is becoming cheaper as well. And people are coming up with lots of sort of creative ideas about producing solar power. So I think it is still an issue, access to computing, but I think the technology is becoming cheaper all the time. And I think there should be sort of funding towards trying to get more access to people that you talk about to sort of computer facilities. I don't know if Ross wants to make a comment on that. No, is he there? Maybe not.
I don't know. I don't think so.
Okay.
It's okay.
Oh,
Yeah.
Ross, he's there.
Hi. Yeah, was Pedro asking a specific question about the computer you used in that
No, it's
example?
talking about the access to sort of computers with the cost of computing, and I'm saying that like school kids, teenagers, say for example in Rio, they're not able to get access. So I think there is an increase in sort of cheaper types of computer equipment that are available, and that's something that we should sort of encourage.
Yeah, so I think it's maybe like some of the open source communities, maybe in Rio that might, or in that might be able to help with that stuff. Like I kind of know a lot of people in Colombia who sort of distribute Linux, cheap reused Linux computers and software. So they're not always having to like buy really expensive computer stuff. So, but it's always a problem, access to technology. So it's, it's cheap for some people, but not cheap for others. And that's always difficult. But I wonder if the open source community can help, is trying to help with that. I don't know.
I don't think they know what the open source community is.
So it's maybe like free software that's not Windows or not Apple Macintosh. There may be freely available operating systems like Linux.
Yeah.
So, which is popular in computer science anyway. I'm not sure.
Yeah. Okay. Good. Thanks.
Right.
Well, we have a question from Carlos Perez. Have you ever thought of joining science, arts, and cuisine? I think he's asking that because of insects, not because of your bread.
I think that is a very interesting part to talk about the positive side of the insects. One is the nutritional aspect that it should be taken into account.
Carlos, don't
I
you
think
want
that
to...
is what Carlos is trying to say. Yeah.
Yes,
You mean like this?
Yeah.
Oh, okay.
So yeah. It's a whole range of recipes and things.
Okay.
So, a very good book.
Okay.
Yeah,
so,
so I used to actually eat locusts in my youth. We had to sort of occasionally we would sort of have a stir-fried locust feast, but we never did it with sandflies.
Well. Well,
we're going to have a problem with production.
Yeah.
Yeah. Well, we have congrats from Silei and from Marcia. Ita Bajara is sending a paper relating music to protein interactions.
Okay.
And well, somebody has more questions or? Well, yes, saying thank you. Do you want to make some remark or
me? So yeah, just thanks very much for joining me for this great talk. Not my great talk, but
Yeah, it was really a great talk. Yeah.
It was a great talk.
I was great
Yeah.
to join. It's now 9:30 in the evening here, you know, so
sorry about that.
So yeah, I've enjoyed it. It's been great. That's what I mean. To sort of discuss a little bit about science, but also about the things that I'm really focusing on now in sort of the more recent years in terms of sort of public engagement and sort of working with other communities. And I think things like sort of working with people who actually are getting these diseases, we should think of them as the experts as well. And because they may have observations about things that we're not aware of, because they actually live with the insects, they live with the diseases. And I think getting people, scientists, young scientists, to actually go and perhaps live in those communities or visit and develop trusting relationships with some of these people is one of the ways forward to actually then co-design and co-develop new prototypes for actually tackling some of the problems that we have. So we are not necessarily the experts, and it's not a top-down thing. It's something where we work together with communities and they are also the experts, I think. So that's what I want you to sort of think about.
Okay. Well, there are more messages. Thanks for the nice talk. Well, Rod, I'm really grateful for your talk. It was a really very nice talk and a very nice conversation. It was a great pleasure to have you with us. So I think it's that. I know it's late there, so.
Oh, late. It's.
Yeah, well, yeah, it's late for a science talk, but anyway, thank you very much for joining us.
Okay, it's a pleasure, and I hope to be there with you physically as soon as permissible. So, yeah.
I'm expecting that too, either.
Okay.
Of course. Okay. Thank
Bye.
you. Thank you,
for a
everybody.
very nice talk. Nice to see you.
Thanks, Rod. Take care.
See you later.
Take care.
Okay, take care.